Hello, my dear students. In this class, let us study about cyanobacteria as biofertilizer. Cyanobacteria as biofertilizer. Cyanobacteria are considered to be the first form of bacteria to carry out photosynthesis. We are capable of converting carbon dioxide to oxygen and to increase the oxygen level in the atmosphere. They are having high ecological significance. They have high ecological significance. They are phototrophic microorganisms, which means they require light in order to get energy and in order to multiply. So they are phototrophic microorganisms. At the same time, there are certain cyanobacteria which are capable of secreting toxins by abundant growth on the surface of lakes and other water bodies. Hence, studying cyanobacteria is highly essential in order to understand their mechanism, their living types, their isolation, etc. Most important is their capacity to fix atmospheric nitrogen, both by free living and by forming symbiotic association with water forms. Example, azolla. So let us see how this atmospheric nitrogen is fixed by cyanobacteria. My dear students, we all know that nitrogenase is the enzyme which converts atmospheric nitrogen, which fixes atmospheric nitrogen or converts atmospheric nitrogen into ammonia. The similar strategies have been implemented by the cyanobacteria as well. But here, as we all know that it is a phototrophic bacteria, they need to carry out photosynthesis as well. So how does these cyanobacteria are capable of converting the nitrogen into ammonia and fixing the nitrogen? Let us see. In order to fix the nitrogen, certain cyanobacteria are having a specialized structure called as heterocyst. This heterocyst lack or have low amount of oxygen level, and the cell wall is made up of glycolipids. In this site, the nitrogen fixation takes place, whereas These are termed as vegetative cell, and here photosynthesis takes place, which means nitrogen fixation via heterocyst. This is one of the form, and there are certain cyanobacteria which are not having any heterocyst, non-heterocyst cyanobacteria, especially in oceans and other marine bodies. During this condition, they are capable of fixing the nitrogen during nights. In the absence of light, they try to fix nitrogen, which means there are two types. One is heterocyst, another one is non-heterocyst. So let us see some of the examples of cyanobacteria. Some of the examples are nostoc, Leucapsa, Anabina, etc. These Anabina can form a symbiotic association with azolla and can perform nitrogen fixation, whereas these are free living forms. Now the next important thing is how to isolate the cyanobacteria. Here it is very pretty simple. The samples are collected from like water bodies and which is subjected to serial dilution in order to decrease the load of the sample. Here, my dear students, here is a important step. In normal serial dilution, we used to have a petri plate. If there is a media present already, then we used to add the sample. It is called a spread plate, and if you have an empty petri plate, then you are adding the sample first, and it is called as pour plate. But in the case of cyanobacteria, the serial diluted samples are inoculated into broth culture with specialized media called as Prinction media. This media contains the essential nutrient for the growth of cyanobacteria. After this, it is incubated at between 25 to 28 degrees Celsius for 4 weeks under the light. light. So after 4 weeks under the light, if you see, there will be thread-like structures growing inside this media, and these are nothing but cyanobacteria. So with this, let's conclude this lecture, which gives an overview of the role of cyanobacteria as biofertilizer in order to fix atmospheric nitrogen. Thank you for your kind attention. I wish you very good luck for your examination. If you have any doubt, comment or call us anytime. Good luck.